Hey, Sam, it's Brandon with Voltage Park. Good to see you again.
Good to see you too.
Thanks for joining me. Who are you? What do you do? Who is Future House?
Yeah, so I'm Sam Rodriques. I run Future House. I'm the CEO, and we are an AI for Science research lab in San Francisco focused on building an AI scientist to
Nice.
automate discovery in biology and kind of other complex sciences.
So, are you a biologist? Are you a microbiologist? What's your role?
Yeah, I'm a bioengineer. So I was a theoretical physicist by training. So I started out doing quantum information theory, and I ended up feeling like there were no unsolved problems left in physics. And so I moved into biology and neuroscience, where like everything is unsolved, and decided that what I really wanted to do was to understand how the brain works. And so I did my PhD. My real like technical background is actually like wet lab bioengineering, figuring out how to build new tools that can get more information out about like where different cells are in the brain, what kind of functions they have, how they're connected to each other, and so on. And that's work I was doing with Ed Boyden at MIT. But kind of one of the main things that I learned in the course of doing that was that even if we had all the information we needed in order to understand how the brain works, we wouldn't necessarily know it because no one has enough time to go and read all the scientific literature. And even if they could, they wouldn't be able to go and assemble it into one kind of comprehensive whole, right? In their brains. Like you wouldn't be able to remember when you got to the end what you read at the beginning. And so that's basically what convinced me that actually the most important thing that we need to do in order to like understand biology and make new discoveries in neuroscience and therapeutics for medicine, everything, is to figure out how to build an AI scientist that is to build something that is better than we are at understanding complex science. And that's the mission that we've been on at Future House over the past year and a half or so.
That was kind of my next question. You're a bioengineer. What business do you have in AI?
Yeah.
Now, are you truly an AI building an artificial intelligence biological scientist or bioengineer? What's the difference between machine learning in science or AI in science?
Yeah. Okay. So this is a great question. So, the first question is, what is the difference between machine learning and AI in science? So people have been talking for 10 years about how machine learning is going to revolutionize biology. And there's one major success in particular, which is AlphaFold, in which there was like a real major unsolved problem in biology that was solved using machine learning. And now there are many other examples of people using machine learning in science. A good example is that like the Arc Institute has these like DNA models, right? They have like Evo, and several other people also now have DNA models. The Evolutionary Scale is another company that is building big protein models, right? And there are many, many other companies, like even those are newer generation machine learning for biology companies. There are many other older companies, you think about like Recursion, for example, you think about like In Sitro. There are like even companies that are older than those as well. The key difference is that usually when people talk about machine learning in biology, what they're talking about is building a specific model to predict one phenomenon, right? So you're building a model for proteins, you're building a model for DNA, you're building a model for cells, or a model like to predict specific aspects of a disease or whatever. When we talk about AI in biology, what we're talking about is something fundamentally different, which is we're talking about building a system, an agent basically, that is able to go and do scientific research the same way that humans can. So that is able to go and do literature search, that is able to go and do data analysis, and ultimately that's able to go and generate new hypotheses, and then maybe even carry out those hypotheses, like for example, by interacting with either robots in the lab or interacting with like contract research organizations or things like that. And so, you can imagine that like this AI scientist, the machine learning models, right, AlphaFold and Evo and all the others are still super important. They're tools that would be used by the AI in order to understand science. Those machine learning models are down here, and the thing that we're trying to build sits above them, right? What's really the difference is like really the layer of abstraction and the underlying technology, right? So in the case of like AI for science, like AI for science has really been unlocked by the advent of the large language models, which is what we build on top of as well. There were certainly prior to large language models, there were other people who are trying to do similar things using kind of more traditional policies. But the thing to understand with the language models is that you can't represent, like discoveries in biology are represented in language. So if you think about like a kind of discovery that you would make in biology that you might read about, for example, in Science or Nature magazines, you could imagine that the discovery would be something like there's like a new kind of neuron in like the vagus nerve, for example, that is like stimulated by some metabolites from the microbiome. And when it's stimulated in such and such a way, like it changes its activity pattern that contributes in some way to Parkinson's disease. I just made that all up. It's not true. It might be true. I don't know. But
I was going to say, you heard it here first on Voltage Park's cast.
exactly. No. I made it all up. But the point is, that's the kind of thing that would like count as a major discovery in biology. You can't represent that in the latent space of a DNA model, right? Like it's not something that you can represent in terms of DNA. It's not something you can represent in terms of like proteins. It's not even something you can represent in terms of small molecules or in terms of like cells. It's like the interaction between all of those things in a very conditional way that's like conditional on environmental factors. That's how biology works. Those are what discoveries look like in biology. And the only representation space that I know of that is sufficiently diverse to be able to like contain discoveries like that is actually natural language. So we think that like language models are actually an essential part of discovery in biology because they're the only things that we know of that can represent the discoveries that need to be made.
So the whole world is really focused on LLMs. They are what's popular. So you have LLMs, you have AI running on Voltage Park GPUs. How does that get into a three-dimensional real world? Do you have robots in a laboratory somewhere trying to recreate something that you've created in the H100 laboratory? But where does the real world meet AI science?
Yeah, so we do two different kinds of things. So the first thing that we do is that we build agents. Building an agent basically consists of building some architecture around a language model. So taking a language model and endowing it with tools that allow it to search the scientific literature, for example, to analyze data and so on. So a lot of people, when the language models first appeared, a lot of people realized was that they like hallucinate a lot and that made them very bad at science. I would ask them, for example, who was like the first author on a paper that I published in grad school, and it would like say someone else random and I'd be like, oh, that makes me very sad, right? Though it turns out that you can solve those problems if you give the language models access to the information that they need in order to answer the questions they're trying to answer. But that requires additional architecture. OpenAI and Anthropic are not building that architecture. And even in the cases in which they build something that looks like that architecture, so for example, deep research, you can use deep research to answer scientific questions, but deep research is actually like not specialized for the scientific literature. If you go and you build the architecture yourselves, which we have done, you can do much better. So we have an agent called PaperQA, which we built, which we open sourced, which is actually capable. It provides answers that are very much like deep research, but it's capable of searching about 50 to 100 times faster and considering 50 to 100 times more sources because of the way that we have specialized it for the scientific literature as opposed to, deep research is like a general purpose thing that will do whatever you want, right? And so that's the first thing that we do is that we go and we build agents that are specialized for science. And when we build those agents, we build them on top of language models from the frontier labs, right? So we use Claude and we use OpenAI's models and so on. That's the first thing we do. The second thing that we do is that we explore actually training our own models by taking open source models. So for example, we work with models in the range of like 30 to 70 billion parameters. And, we've gotten a lot of mileage out of the Quen models, for example. And we go and we then train them also to do like specific scientific tasks. And it turns out that if you spend just a little bit of effort training your own model, even a relatively small one, you can beat the frontier models very easily. And so that's the second thing that we do. And then obviously you can put these two things together. So you have an agent running on frontier models. You have your own model that you've trained. You can put them together and train your own agent. And one of the things that has been very cool about working with Voltage Park is that we have enough compute actually that we're even able to like go up and start to train like the really big models like DeepSeek V3, the full scale one, 670 billion parameters. That's the kind of thing that we're able to do with the help of Voltage Park.
And so you go from the digital world and you're fairly large in Voltage Park and a superpod at this point. How do you bring that into the real world?
So then we have our own wet lab in-house, actually. At the moment, we have our own wet lab
in the
in-house.
Office.
Yeah. Can I show you?
I would love that. Can we show it?
Yes, definitely. Well, you can just see part of it. And the part you can see is really cool. So it's bigger than what I'm showing you.
Yeah, show us the cool part. Whoa.
So you can actually see the entire, so what you're seeing is we're in this warehouse and that structure that you see is the wet lab with all kinds of, with the equipment inside of it. And you can see the ventilation on top.
That looks like, 1,000 square feet, 2,000 square feet of wet lab.
Yeah, that's 2,000 probably, and we have more than that. I mean,
What does that mean for the digital world and then the wet lab? Where do they come together?
We literally have our agents spitting out hypotheses and proposing experiments, and then we have people who are in the wet lab and we go and run them.
Cool.
And we have some 3, 4, 5,000 square feet of wet lab space, which is enough for a pretty substantial wet lab operation. When we need more, we need specialized things that we don't have here. It's very easy to just outsource it.
What's the success rate and the failure rate on something like that? So for like material sciences, it's very similar where they're trying to create a new material and AI is accelerated in harmony with
You would
ML.
be shocked. You would be shocked. So far, we have models spinning out hypotheses, and we have gotten success rates in our early experiments on the order of like 40%.
Whoa.
Yeah.
That's amazing. What does that mean for human biological science? What roads are we on? Are we trying to reverse aging? Like,
Well, let me put it this way: the
get
success
rid of
rate,
Parkinson's, get rid of cancer, get rid of MS? What are we doing?
the success rate currently for drugs, like the overall success rate, like you have a drug starting at the beginning, and how likely is that drug to get approved is like 7 or 8%. Okay, if we could even like double it, you would see the times that take to cure diseases get cut in half. And you would also probably see the prices that medicines cost get cut in half, right? I mean, people have to appreciate that the cost of medicines is primarily driven by the cost of R&D.
Right.
And so if we're twice as successful at making medicine, it will bring down the price, and it will also bring down the timelines. One of the things that people really have to appreciate is that the vast majority of discovery in biology today runs on serendipity, right? Serendipity means that there was an idea that was sitting, waiting to be discovered, that wasn't discovered because no one stumbled across it, right? So one of the best examples of this is CRISPR. CRISPR was discovered in 1987. It was not appreciated that you could use it to edit genomes for like 20 or 25 years after that. How do we get to the point where rather than waiting 20 years to have gene editing technology, that we are able to, like, as soon as something is ready to be discovered, that we discover it? Serendipity is a very romantic concept. It's a very beautiful concept. You get to tell stories about how you like gather together, two people meet at like the water cooler and they talk, and then they have an idea, and that's what drives science forward. That's amazing. It's also bullshit that that the progress in science is waiting on people to meet at the water cooler, right? We should be doing better than that. And I think we can. And the way that we're going to do it is by having these models systematically go through what are all the possible hypotheses? How do we sort through them, surface the best ones? In 1988, we should have known that CRISPR could, had the hypothesis at least that CRISPR could be used to edit genomes. How do we just make sure things happen as soon as they can?
That's fantastic and fascinating, because serendipity will actually go away, and it will no longer be timing and luck. It will just be pure horsepower thrown at it.
Yep, that's exactly right.
Is there any projects that you can tell us about that you're excited about that is going to be public?
Yeah, for sure. So one of the kind of really exciting examples that we've been exploring recently is we've actually been just exploring to what extent can these models come up with new treatments or can propose new treatments for diseases. And we had a case recently in which we went to the hypothesis agent and we said, can you come up with a new way to treat age-related macular degeneration? This is a major cause of blindness that is associated with age. The agent went and at first identified, how would you know, or like what experiments could you run in order to test whether something is effective for treating age-related macular degeneration. And then it was able to go and come up with a specific molecule that would use in this particular assay that it came up with. One of the key factors of age-related macular degeneration, at least in cells, is that they have reduced phagocytosis. That means that the cells are less prone to eating stuff out of the environment. And so it said, how do we go and we improve phagocytosis? It found a molecule that has never been studied before in the context of AMD. We take that molecule, we put it into on the cells, and it worked. And it worked really well. It actually worked better than the positive control, right? So that's very exciting. That was week one. On week two, we got 3 more hypotheses that also worked. So, this is what I mean when I say things are just going to move much faster. But it's not just that, which is that then once we've got the result that worked, you go back to the model and you say, hey, that worked. What should we try next? And the model then proposed something that is actually an already FDA approved drug that is not used for age-related macular degeneration. Again, it's not proposed anywhere, not talked about as a treatment for AMD. You should use this. We take that, we put that on the cells. It also works. It actually worked even better than the first one. So now we have a very, very clear path for can you just repurpose this existing drug for AMD?
I think everybody's probably thinking right now, using it for negative and malintent. What are the ethical guardrails being put in place in your industry, if there are any?
Yeah, it's a great question. I think the thing that you need to appreciate is that we already do have certain controls in place at the level of actually like performing the experiments. There is some screening procedures that are put in place when you try to order DNA to prevent you from ordering things that are known to be very, very dangerous. And there are a lot of people who are thinking about how we improve the quality of those controls, because with AI, you might be able to like figure out how to circumvent them or whatever, right? And I think that that's very important and very good work and should definitely be supported. So maybe the key thing to recognize is that there's always a bottleneck, which is that in biology, you have to actually do the thing. In cyber, for example, if you think about like the cybersecurity risks of AI, actually it can just run on your computer. It's much easier. The barrier is probably lower. In biology, you have to have access to a wet lab. You have to know what you're doing in the wet lab. Maybe the model can help you to figure out what you're doing, but it's hard. And so that's like the first kind of key barrier. On top of that, we do just need to, in the same way that the big model builders think about how do we prevent people from using these models to like make bombs or conduct human trafficking or whatever other terrible things people might think about asking these models to do. I think we need to think about how do we make sure as we make these models publicly available, the agents that we're making publicly available, how do we prevent them from being used for nefarious purposes? To a large extent, because we work with language models, the agents that we're built at the moment are built on top of language models that are being produced by the large model builders. They have safety procedures already in place, and we work with them very closely actually on thinking about biosecurity and biodefense. We work with several of the large model builders on thinking about that.
That's good to hear.
It's an important problem.
It is obviously very difficult, and there's not many people lucky enough to have the talent that you have, access to the GPUs and access to a wet lab right there at this exciting time. What do you think is going to happen in the future of hardware as the GPUs get faster? Do GPUs getting faster and more efficient help your business?
Honestly, I don't know yet. They help in terms of allowing us to train better models on more data. Absolutely. Same as everyone else. The thing that the caveat that you have to appreciate is that also in this business, we will ultimately be limited by the wet lab, right? Which is that the models will be proposing hypotheses, and the models will get extremely good at proposing hypotheses, but we will be limited by how quickly we can test them. So the problem is as much logistics as it is AI, right? It's like, yes, the AI, is how do you come up with the best hypotheses possible. The logistics is having come up with the hypotheses, how do you test them in the most efficient way possible?
Yeah, very similar to material sciences, right? You can come up with all these new compounds, but until you get into a robotics lab and try to create those compounds to do various different things, it's putting the hypothesis into production. And what is your success rate on that? And how do you increase your success rate? 40% is amazing. What's your goal?
Yeah, the thing that you have to understand about biology also is that when I say 40%, this is an assay that we ran. The hard part about biology is it is in fact the case that you can run the same experiment twice and you can get different results. And for reasons that you don't understand. And by the way, for the record, the same thing is true in machine learning. People don't like to talk about it, but you can definitely train the same model twice and get different results.
Yes, yeah. It's got a mind of its own. It's weird.
Yeah. And so, this is science. And so, look, the jury is still out on what the science is ultimately going to look like. And in terms of like what we're looking for, like what success rate do you want, like whatever, I have no idea. It's just too early to say. I can just tell you that already we're in a position where people should not be writing their own literature reviews anymore, right? Like I can just tell you, like paper QA is better. In most cases, paper QA will be better. Provide you with a better summary of the literature than anything that you're going to go out and make yourself. That's a no-brainer. Give it 6 or 12 months, and the same thing will be true of data analysis, right? You're going to be conducting data analysis on your own? No way. Not a chance. Why would you do that? Claude can already program 10 times better than you can, right? And so, the
idea... Cursor, and they're in competition, which is just going to make them go faster and get better.
Right.
Competition is good in this industry.
Right. And this is certainly true for people who are, for scientists who are doing data analysis. Maybe Claude is not necessarily yet at the level of the top, like ML engineers, software engineers, et cetera, but just give it some time. So what is it going to look like in 3 years? I have no idea.
Well, I wish you all the luck to you, your team, and Future House. We hope to continue to be a part of accelerating something that's making a difference.
Yes.
I've been in IT for a long time, and the interesting part about being a compute provider is you get to be part of the building blocks of people in microbiological research, in material sciences, in cybersecurity, things that are actually making a difference in healthcare, drug development. This is just really awesome to know that the blinking lights inside our data centers and those big GPU clusters are being used for something that maybe my kids can benefit, or even myself can benefit for one day. So just want to thank you for that.
Hopefully you yourself.
That's right. Yes. Yeah. I'd like to keep my hair. It just keeps receding. So if you can work on that for me.
We'll put it in. Thank you so much.
Pleasure. Sam Rodriques, amazing seeing you again. We wish you all the best and all the luck, and we look forward to your product launch.
Thank you.
Yeah. Have a great day.
See you.